Hello, everybody. It's a pleasure to be presenting to the Bioprocessing UK 2022. My name is Leonardo Rios. I am based at the University of Edinburgh and the Center for Synthetic and Systems Biology. But then also recently I will be moving to the university in April, so very soon, University of Newcastle. So just to let you know, for further updates, if you want to be in touch with me, I will be moving to the School of Natural and Environmental Science, and I will be moving there as a senior lecturer. So quite excited about that. So, well, I mean, what my group is really working is in the interface of synthetic biology, where we see a little bit in the left, like uh, using engineering principles to design uh, biosystems, and especially in my case, microbial cell factories. But we combine that also with bioprocessing technologies. Can we scale up those systems in a sustainable and also economical way? So, so then that's a little bit the two expertise of my group. And so, yeah, that's what the name of this talk is, leveraging how can we achieve this merging, and especially using microscale tools, microscale bioreactors to quickly, let's say, design those systems and then scaling them up in a rapid way. And this talk, we're going to focus specifically about some work we have done with an anti-cancer drug called Taxol, which is one of the most widely used natural molecules used for curing many cancer diseases, but then still not produced in a very sustainable way. So we will find that we would love to produce it using microbial cell factories, but also in a cheaper way. So as I said, in my group, what we do first is we develop interesting natural products from plants. Like in this case, is anti-cancer drug Taxol, which it was not us who discovered that, that's been already previously discovered. But then how can we produce it in a sustainable way? And ideally, some of the tools that we are developing, some of our methodologies to scale up the production of those systems will help to reach a circular economy where we don't produce any waste. And and then those same tools could be applied to, let's say, other type of molecules like biofuels, where the market is a lot bigger and it can really have an impact in this area. And then, okay, going back to Taxol. So Taxol, is from a tree called Taxus, many different Taxus species. We can find them easily here in Scotland. You may have seen them if you are from this area with those lovely little berries. So the Taxus is mainly produced in the needles of those trees. Before, they used to take the tree, the whole bark, and they had to get lots of different trees to produce a single dose of this drug. So they almost got extinct sometimes those trees. Now there are a bit more advanced methods where they use the needle and they extract an intermediary of the Taxol, and then from a chemical synthesis they obtain the Taxol. So it's a semi-synthesis, still relying on natural sources and semi-synthesis. But we would like to make it totally not have to rely at all on the trees and produce it in a microorganism, in this case, S. cerevisiae. And so part of the first work, which we're not going to go very deep in this case, but that's part of also part of our team, is how to build these microbial cell factories. You can see there the whole pathway, and we haven't actually reached the full Taxol molecule yet. There's still steps to discover to this molecule. We are almost in the 10 beta hydroxylase state. And as you can see, some of the genes in green have been overexpressed. They come from the plant, they have been overexpressed. Some of the genes in orange, we use different microorganisms. Some of the genes in red, We have put several copies of it, and yeah, we're excited. We're one of the groups that have reached one of the further steps using that microbial cell factories. And then, okay, we were ready to start scaling and try to produce those intermediaries in a bit larger scale. And so, how did we proceed to do that? So that was one of my PhD students, and now she's a postdoc in my group. Laura was her expertise was how to use different microscale bioreactors to rapidly optimize those systems. After we build them, how to see all the effect of the different bioprocessing variables in those systems. But we want to use them in the small scale tools, but that they can mimic larger scale bioreactors. So some of those examples are here. We can use some microplates, but that they have integrated sensors, and that the shape of the well will give enough oxygen, for example, or will mimic the fluid dynamics of a larger scale system. And then, okay, we have also some mini bioreactors here from Applicon, 250 milliliters ones, but they totally mimic really well. They have all these impeller, shear stress. They mimic quite well a larger scale system. So we wanted to leverage those tools to see, how valuable it was that to quickly optimize those bacterial cell factories. Laura's proposed approach was first to do some trial experiments in shaped flasks, so not start immediately from a smaller scale and going to larger scale, but first some trial experiments in shaped flasks to test some extreme conditions, some non-extreme conditions, and see how that's affecting our cells first. Because after you get this quick information, then yes, we use design of experiments. To vary, to see the effect of all the process variables, including pH, including oxygen, including shaking speed, including inoculum concentration, including all the different media components, including temperature. In addition to, we could also put different strains, the different genome, engineering strains that we have done. So that adds quite a lot, So that's what we do some trial experiments to see, and then yeah, we do the design of the experiments. We're not gonna go deep into all the design experiment, all the optimization that was done here. Just highlighting this methodology that worked really well. After that, okay, then we did the scale up in some of the larger scale variables. But the type of data that we would get here, we're showing a little bit we could start to see some of the bottlenecks of the data. First, we could see the biomass, how depending on the different media and components, we had higher or lower biomass. We wanted to go higher. We could see the pH. Usually pH is totally neglected. We could see how the pH changes were totally affecting the whole pathway, and we could see which pH was affecting more one type of enzyme and not another enzyme. We have different enzymes from different microorganisms, so the pH has a different effect on each one of those. So it was about finding a trade-off, so you want to affect maybe the one that is the one to affect less, the one that is the key enzyme, the bottleneck of your pathway. And then after you relieve that one, you may see which one is the next bottleneck and go into that, Oxygen, dissolved oxygen was also very interesting for us to see how it was actually critical, not only for, our yeast didn't need it, so oxygen to grow, we wanted to keep it in a fermentative metabolism, but it was actually more critical for some of the enzymes that we're talking about P450s were doing. They needed some oxygen, some of them, and that was key to really also improve this pathway. So, and then, okay, what we did in the bottom is the metabolomics. And we saw that the process variables had a huge impact of the metabolomics profile that we were getting, especially because when you're putting those pathways from plants into yeast, for example, you may get many side products and unwanted products. And then those different bioprocessing variables have a big effect on the amount and type of unwanted products that you want, so the idea is to minimize those and get the product specifically that you want. So after that's been successfully done, the advantage is you can model also that too. We could model both biomass, both the software consumption, ethanol, and also the product production. We could model it to a certain degree. We can get the coefficients of product to biomass, product to substrate, and that's good enough for us then to do calculations, scaling up, and or also starting to think about different type of bioreactors. And that's when we realize, in our case, a continuous flow bioreactor would be the ideal based on this model. It was telling us our production product is linked to the energy metabolism. It's continually being produced with growth. Continuous flow would be the best way. And so that was what then, after getting this information, is how to design this continuous flow bioreactor. That was a bit the task of my other PhD student, Jorge Santoyo. It was okay, that's how to do an integrated bioprocessing where we can continuously grow the cells and produce our product, but also at the same time we needed to continuously remove the product. I mean, it's fine to keep growing the cells continuously, but if you cannot keep recovering the product continuously, then it's useless. So that was the task of Jorge, how to achieve that. Normally, the way we were growing and removing our product is we're using an organic overlay into the, we have our water with our cells. We were using dodecaine on top of it, so we could then remove our product is totally hydrophobic, so it will go to the dodecaine, and from dodecaine we will purify our compound. But that's environmentally not friendly, and actually to produce our compound, to really purify our compound from dodecaine was extremely difficult. It's very difficult to evaporate dodecaine. So, and then when you go into a continuous flow system, mixture of water with dodecaine is impossible really to keep continuously separating. So Jorge had the idea then to use a solid liquid extraction. So using beads, in this case he selected to use the HP20 resin bead. We actually tried other beads, but we got very interesting data that we may patent on other results, so we couldn't show some of the results with other beads yet. And that was exciting, but we can show the results of that one, that this work has been published recently. And the idea is you have the yeast, you feed them, in this case, galactose. Now we have strains that can grow on glucose or raffinose, and it has a whole engineered pathway inside. But when it produces taxadine or other different intermediates, we have to select a bead that will selectively absorb the taxadine. And so it will allow us to work in a bioreactor setting like this one. We're controlling all the bioprocess parameters that Laura obtained, but we're growing the beads at the same time first as the yeast. So that's a preliminary step before moving to continuous flow system. First, you want to try just that your in situ method to remove the compound, that it works, and you want to optimize it first. So Jorge did that. First he developed the fermentation step in the left, he's growing the cells with the beads, but then he had to develop a whole method to extract like the products from the beads, for example. So it's this whole thing, like growing the cells, but then the beads are easy to recover from the fermentation system, and then you need to find a way to recover in this case that intermediates of Taxol. So he did that, but first he tried to test, okay, the concentration of beads, does it have an effect if we're growing the beads with the yeast? And see, we have a graph where we try different concentration of beads, and we see the growth of the microorganism with time. We can see they grow fairly well, except with the dashed line, which is after 12% of resin beads, the cells, they totally died. That was in the microscale, that was in the shake flasks. And so after further investigation, we discovered that the beads, they were actually, you see in the left, that's when we started the fermentation, the beads are actually quite big in comparison to a cell. This is a quite big one, and the cells, the yeast cells, actually quite small. But when you start doing the fermentation with time, with impeller, or just shake flasks, which was in this case microscale, the beads start to break and they create some small little fragments that kills the cells. They break the cells. They're quite actually small. They work as needles, so they were killing the cells. So then, yeah, if you put many of those beads, there is a balance. You will then totally crash the culture. So you cannot add more than 12% without, like having killing the cells. So, but okay, we saw that okay, it works with 3-6% beads. So that was fine. And then okay, then we studied, like where is it really all our taxadine and Taxol intermediates moving to the beads. So in this case, we separated the liquid phase from the solid phase, and there was almost none taxadine and other how you call it here, like Taxol intermediates in the liquid phase. And in this case, it's important to say the solid phase was the cells plus the beads, so they were both together. It was just a separated liquid and versus solid phase. And we got so that everything was in there. But things could be, the product could be in the cells or it could be in the beads. So that was also a question that we were not so sure about it. So then in this graph, as you can see in the middle, you have how much of the taxadine was staying in the beads. That's the middle one left. And then the middle one right is how much was it staying in the biomass. And then you can see in the right, the gray one is the total one, beads plus biomass together. If you say, normally the beads plus the biomass, it should match, it's a mass balance, it should match when the one of beads plus biomass, But then when we were separating the beads and the biomass, there is inherently some, some steps, an extra step, and some of the taxoline is easy to evaporate. Some of it is lost, you cannot clearly separate everything so nicely. So there are some losses, and that's why we're not getting the exact mass balance of how much is in beads plus the biomass should be equal to the bigger gray. But the key thing is to say that it was half almost of the taxadiene was in the beads, half was still in the biomass. So that was a key, very interesting thing to see. It means that not everything was totally in the beads, And interestingly also to say that we produced a lot more, almost a double, more than a double, that we were using our dodecane system. So this method was a lot nicer. It is less toxic. Dodecane is a bit toxic to the cells. This was less toxic, and we were quite happy to see it was actually a lot more efficient producing than the red one, the orange one, dodecane. So then, okay, then we developed just a method to show how to extract. Now we recover it from the beads, but how are we going to totally extract the taxadine? We try different organic solvents, ethanol, acetone, ethyl acetate, dodecane, the same one and we actually saw that using acetone was a lot better than the traditional method, using dodecane or ethanol acetate. And that was because it was a trade-off. Acetone was very good to extract from the beads, but it was also breaking the cells very nicely and recovering all the taxadine inside the cells. So it was like a two-step process just in one go, breaking the cells to remove the taxadine from there and also from the beads. So that was a fantastic for an integrated bioprocess, So then that worked really well. We tested also, we wanted to see, okay, we're using in this case 3% of beads. Can we go lower? Maybe 1, maybe 0.1%. Beads are a bit expensive, So how much can we go lower? And we were okay, interesting, pleased to see that you can actually go to 0.1. And you can see in the right, the growth, there is no effect at all in the growth. This is the growth of the cells with different concentrations from 3 to 0.1, no statistical difference. Production rate, 3% was still better than 0.1 or 1%, but not drastically better. We're talking a little bit that was almost the double. So that will totally depend, of the value of the product, of the techno-economics. We've seen that the more beads we have, we push to have better recovery, but if the expenses of the beads is critical, you can go to very low and still get a good recovery of the yield. And then, that has to do with many other things because we then discovered, but that's something that we cannot show here, that we were surprised to see that some of the beads have other more beneficial effects. Not only they were recovering taxoline, but they were also helping the cells to be more healthy. And we investigated that. We have other beads that promote that, but that's still confidential. We cannot say that. But that was interesting to see that the beads can have further advantages such as just helping you to purify the compound. So those are very great news, and we will be happy to present those things after we have protected them, and we have also published that new data. In this case, we can only show that the effect of this specific bead. But then we proceeded to scale up in a fermenter. We were quite pleased to see that the results that we were getting in a small scale, they were even in our case, even better. So we have the microplate versus bioreactor. We were consistent and even a bit better in the larger scale. It was actually quite nice to see that in using all the optimized pH, oxygen conditions. So that worked very nicely. So that was really good. We were happy to get that and publish it. And then the next step, in collaboration with Dr. Simone Dimartino from Edinburgh University, we moved towards the continuous flow production of this system. As in this case, and we were been thinking about putting the beads in a column. The idea is put the beads in a column so that we will be recirculating then the cells in a column, and but then it's going to be like a packet bed column, so the beads won't be into the solution with all the mixers, so then they won't be breaking apart, killing the cells, and that would totally does the transition to go towards continuous flow production system. There are challenges to be there. So, for example, the column, depending on the flow rate, the column will get packed and it can get blocked, depending how much the bead type that you use, the density and the flow rate. The column will expand and then get blocked. So there is some optimization things to do there. And so then also this is also a bit work in progress. There's a few things that we cannot show yet because it hasn't yet been published, some of those. But some of the very interesting results that we have shown that I can show here is, for example, in this graph, we're again testing the concentration of beads in a higher concentration, like 19.2 was the concentration of grams. That was the concentration of 12% when the beads in a batch, they were all dying. In this case, when we put them in not recirculating, but in the vessel, that's the blue line. So then the cells, they totally die, same as it was happening when we were going to check flask in a bioreactor. And the right is the galactose, they consume. But in this case, we can put a higher amount of beads into the bioreactor if it's recirculated, and the cells, they won't die. And then the cells, they will actually grow very nice, and actually they will grow even nicer than the control, which is some of those beneficial things that I've been talking to you, that we were quite surprised. We've been finding out what are all those benefits that, and so still confidential, but it's that we can really, it's very interesting, and it also only helps to remove the compound of interest, but it has the cells to be healthier. So then running those systems, we got, for example, the 12% in the middle. For example, if it's the 12% ERCs, it's the internal, like growing, putting the beads inside the system, like shaking with the beads and the yeast growing at the same time. So they die. They don't produce almost anything. 3% is what we were doing a bit before. We were, a lower amount of beads in a bioreactor. We were able to produce some of those in the cells, some of the similar amount in the absorber. But with the recirculation system, we could go a lot higher in the concentration of absorber, especially. And in this case, actually, the balance shifted that the majority of our product would stay in the absorber and almost nothing inside the cells, because we could really then put a lot more concentration of of the absorber. So that was actually very promising because it helps us to purify it more easily, and we can go a lot more aggressive in the amount of bits that we can put, and then that's enable us, to develop then those continuous flow systems that we are now finishing, and hopefully we will present hopefully next year after we can publish all the latest very interesting results about that. And then to finishing with this, we just want to conclude, that we're totally convinced that integrated bioprocessing will be crucial for the future of biomanufacturing, especially towards this shift to continuous flow. We're totally convinced that this will really lower down the costs by at least one-fold, to tenfold, and tenfold, yes, and then that will really make many things economically viable using biotechnology. But we wanted to caution that characterization of all those individual units of operations, for the extraction and the growth of the cells and the different condition parameters. It's very necessary. It involves lots of different experiments. So using microscale tools, using this kind of experiments is quite necessary. And we were quite pleased to see that when you are integrating those upstream, downstream, the downstream processing can have a radical positive effect on the upstream section if properly integrated, and that can be actually a lot very interesting, to even improve the economics of the process. What are some of the then also challenges that we are now tackling, or at least characterizing, is for the continuous flow system is the kinetics of the mutation rates of our synthetic pathways, one of the bottlenecks that the continuous flow system have. So can we minimize those? Can we, or at least predict them, so we can find ways to minimize that? Is some of the work that we are doing to future work, to be able to really be able to change this future manufacturing. So finishing with that, I would like to acknowledge all my PhD students, postdoc students, and this work, especially in this case, Laura, Jorge were fundamental, but Benas, Corey, Jorge also participated a lot, and especially also Rafael from the Simone's group participated quite a lot. Funding that we have got from different sources, BPSRC, EPSRC, Global Challenge Fund, and collaborators from University in Mexico, Imperial College, Berkeley, Denmark University was critical for this work, and many other support. Thank you very much.